Let's work through the process of labeling an actual Western blot. We'll use the image from Figure 2C of the Soulier and Lowndes paper in Current Biology 1999 for this exercise. Let's look at the image components we have. As you can see, there are two images. They represent an STS-page gel which has been transferred to a membrane and probed separately with antibodies against the Rad9 protein and the Rad53 protein. The separate visualization of the two different primary antibodies gives us our two separate image components. However, as the images are currently displayed, it would be impossible to decipher any information from them. And so this screencast has been designed to show you how to go from the raw images shown here to a figure that is of publishing standard. First, we must arrange the images in an appropriate manner. The lane order is the same in the two images. Therefore, we need to line these up perfectly, one underneath the other. The initial step in labeling is to place lane numbers under each of the lanes. Since the figures are horizontally aligned, we only need to do this once. Although not obligatory, lane numbers make it easier to refer to any particular signal in the figure. They are especially useful in project reports and theses, where we discuss details more intensively than is possible in a brief research manuscript. Note also that figure part C is clearly indicated at the top left of the panel area. Next, we need to give an indication of the size of the bands along the vertical direction. In a direct gel visualized by staining all molecules, we would normally include a marker ladder in the first or last lane. Examples might be an agarose gel of DNA stained with iridium bromide, or an STS page gel stained with Kumasi blue. In the case of the Western blot which we are dealing with here, the specific antibody means we cannot directly visualize the marker ladder. However, we can usually estimate the separation size scale for a Western blot indirectly. For example, we can measure positions with a ruler and relate this back to the original STS page separation, which we have subsequently blotted and probed. This allows us to place a scale marker on the right for each of these two images. Note also that we clearly mark the unit kDa for kilodaltons. On the opposite side of the images, we also provide some interpretive labels to make the figure more self-explanatory. In this case, we label the phosphorylated and non-phosphorylated forms of Rad9. Note that we use clear lines or arrows to point to exactly which band we mean. Finally, we need to indicate the differences between the samples in the different lanes. First off, we show that the odd-numbered lanes were control samples which have not been treated with UV irradiation. Therefore, we place a minus sign above these lanes. Likewise, the even-number lanes are for samples which have been UV irradiated. We place a plus sign above those lanes. We also clearly label this row of symbols UV to indicate the nature of this treatment. Secondly, each of the samples are paired for minus and plus as control and UV treated. To indicate this pairing, we place a straight line above the minus/plus to link them. On top of this line, we place a label indicating the distinctive property of the strain. The clever combination of symbols is an extremely clear and efficient way of indicating the origin of every sample. We now know that the side-by-side pairs can be compared. Note the use of standardized terminology in the labels. The large P from the atom label for phosphorus beside Rad9 and Rad53 on the left indicates that these are a phosphorylated species. The delta triangle for Rad9 at the top indicates that the Rad9 gene is deleted in this strain. The F and W superscripts for Rad9 respectively indicate the phenylalanine F and tryptophan W mutants using the standard single letter amino acid code. Finally, note how all the labels are perfectly aligned horizontally and vertically with their related neighbours. This attention to detail significantly improves the readability of the figure. Here I'll compare the figure that I have just created with the figure published in the completed manuscript. And as you can see, with a little effort, this standard of presentation is very achievable. So the key points to remember are: arrange images together if related. Lane numbers make it easier to refer to specific signals in a figure. Indicating the size of the bands is essential. Labeling specific bands of importance will make the figure easier to interpret. Plus and minus signs are used for highlighting a specific treatment. Straight lines over markers are used to indicate a group.